Okay, so it's just 3:30. We'll just give people maybe 1 or 2 more minutes to trickle in. Okay, I guess we'll go get started, Mary. Sound good? So I want to thank everybody for joining us today on WebEx. I know it's a little funny if this is your first time, you only see a few of us, but there are about 80 people in attendance so far. So thank you all for joining us. I'm just going to say a few words about the seminar series. This is the second in our New Frontiers in Biomedical Research series for this year that has a focus on impacts and outcomes of a pandemic. So what you'll see from the series this year is a focus on sort of what we've done and where we are and where we need to go and what we've learned, where research should go from here, and what our role might be in all of that. There is a Q&A box in your WebEx platform, so please feel free to type questions at any time throughout the seminar, and our speakers will probably take about 30 or 40 minutes, leaving us plenty of time for those questions. So please just type them in as you have them, and me and Dr. Calderon-Moore will be sure to ask them as that time approaches. So with that, I'm going to turn it over to Dr. Calderon-Moore to introduce our speakers.
Yes, I have the great pleasure of introducing our speakers today. We have a great group from different research areas and backgrounds. We have Dr. Byron Crawford, who's the professor and chair of pathology at Louisiana State University Health Sciences Center, Shreveport. We also have Dr. Rodney Rohde, who is professor and chair of the Clinical Laboratory Science Program in the College of Health Professions and associate director for Translational Health Research Center at Texas State University. And Dr. Krish Roy, who is the Robert A. Milton Chair Professor at Georgia Tech Institution. He's also the director of the NSF Engineering Research Center for Cell Manufacturing Technologies and director of the Marcus Center for Therapeutic Cell Characterization and Manufacturing and director of the Center of Immunoengineering at Georgia Tech University. As Dr. Newman said, our speakers are going to speak in total about 40 minutes, and then we'll open it up for Q&A. So please keep those questions coming as you have them. With that, Dr. Crawford is going to kick us off.
All right, can y'all hear me? Okay. Hi, I'm Byron Crawford. I'm the chair of the Department of Pathology and Translational Pathobiology at LSU Health Shreveport. I also work at the Ochsner LSU Shreveport Hospital, and I have nothing to disclose other than my relationships with LSU and Ochsner. I'm having problems with advancing.
Oh, you have to share your screen, Dr. Crawford. We can't see anything.
Okay, I thought I had. Let's see. I got to find my.
There you go.
All right, I'm going to talk about the road to COVID testing here in Louisiana as it started back in January and February. First, I just want to make a profound statement that laboratory diagnostic testing is the most important component in handling any infectious pandemic and remains so regarding the COVID-19 pandemic prior to a development of a vaccine. In February of this year, the CDC and State Board of Health labs that are here in the state were providing all the testing available for patients that potentially had COVID. To get a COVID test, you had to have a negative respiratory viral panel, a panel that tests for about 20 to 25 different organisms. Most of them are viruses, and if those were positive, then they would not test for COVID. You also had to have a negative strep screen, and you had to be symptomatic. Your doctor then, if you fulfill those requirements, they would contact the physician at the State Board of Health, and that physician would decide whether that you were going to get a test, either it'd be performed in Louisiana or be sent to the CDC. As a result, the state provided very few tests initially for patients that potentially had COVID. This resulted in a delay in diagnosis the first 6 weeks of this pandemic. So non-COVID patients, when they were coming into hospitals, emergency departments, they were also being seen and treated along with the COVID patients. There's great fears at this time of what COVID could do. There was people were talking about there'll be over 1 million deaths in the country. Physicians, healthcare workers were going to die as a result of this disease. So there's a lot of fear going around. And PPEs, personal protective equipment, started to become very limited, not only in the state of Louisiana, but throughout the country. This went on for the month of February. On February 29th, an article in the Wall Street Journal stated that the FDA was going to start allowing other labs, particularly academic labs, to test for high complexity tests for coronavirus. And they suggested it was going to be immediate, like we're going to start testing patients. We're going to be able to get results within the next few days. However, that's what the article said. It just didn't pan out to be that way. At that time, I was in a very unique position. I was still the Associate Dean of Academic Affairs at Tulane University School of Medicine. I was the medical director of the Tulane Medical Center Pathology lab. I had close ties to University Medical Center, which was run by LSU in New Orleans, LSU School of Medicine in New Orleans, and I also had ties with the Ochsner Hospital in New Orleans as a faculty member there. Also, at this time, I'd already tendered my resignation at Tulane to start at LSU Shreveport the 1st of May. So I had contacts with all the major medical centers in the state, very close contact with them, and also with the major healthcare providers in the state. At this point, at the end of February, I contacted all my current bosses, my future bosses, and my current colleagues and future colleagues. I thought would be interested in trying to get set up testing as soon as possible. And I was mainly concerned about the hospital, but in the medical centers as a whole. And I got some very positive response to move ahead with it, but I also received some negative responses, like let the state continue to run the test, or what we're going to wait for commercial instrument companies or commercial labs to develop testing. And we predicted that would take at least 6 to 8 weeks before additional testing would be available. And even one person said it was too dangerous to do testing, which is untrue. We were already testing potential COVID patients and COVID patients for respiratory panels for other viruses, and some of those would come back negative. Sure, they had COVID. And any microbiology lab in a medium-sized hospital has a bio level, safety level 2, and that's all that is needed for testing for COVID. However, the PhDs in Louisiana rose to the occasion, both at LSU and Shreveport. Researchers decided they were going to start develop COVID testing. There's a group of Tulane University researchers. There's two virologists at the vet school, at LSU Vet School in Baton Rouge, who spent their own money to purchase PCR equipment, and they started working on it through CDC to develop their own testing methods. That sounded all great to me, but I was more interested in having it in the hospital setting. New patients are going to be coming in and demanding testing, and also that the physicians were going to demand testing. But for the first week in March, I thought, well, surely we can try to get something started, but it just didn't seem to have much interest with some of the pathologists, and it started looking kind of dismal for the hospital setting. But I knew that I knew the PhDs were setting up labs, and we'd at least have access to those labs. The device, all of a sudden, there's a big surprise around March 15th. It came out that national commercial companies like Abbott and Roche, they were beginning to release kits to the areas of the country that needed the most. The states of Illinois, Massachusetts, New York City, California, and the state of Washington were getting kits. Initially, they only sent out about 150,000 or kits for 150,000 tests, but that ultimately got up to about 500,000 a week by the end of March. So I thought, well, we have an opportunity. We had a Roche COVID equipment in our lab, and I thought, well, maybe we'll be able to get some of the kits. I was told that we probably needed to go through Tulane government relations. They stepped in and worked with our state senators, not our state, but our U.S. senators and our governor. But after about 3 days, it didn't seem very fruitful, and I got depressed again. It looked like we weren't going to get any kits at all. I was on my way up to Shreveport to look for a place to live, and my wife was driving, and the phone rang. It was Dr. Gordon Love. He's the chair of pathology at LSU in New Orleans, and he was calling and says, Byron, I know you have an instrument to do COVID testing. We're supposed to get an instrument, but we won't receive it until July. We all do the testing for University Hospital. That's the old charity hospital. And I said, Gordon, we don't have kits, and we've been trying to get them, and they're just not available. And he said, well, if I can get you the kits, will you run them for, use some of them to run on our patients? And I said, Gordon, but he also said, we have connections that go to the White House. And I said, Gordon, if you can get us kits, I'll split them with you. Half of them will be done on your patients and half will be on ours at Tulane. Within 6 hours, I was notified that we were going to get kits to run 200 tests per day between Charity Hospital or the University Medical Center and Tulane Medical Center. That was on March 20th. The next day, River Road Testing Lab, that's the two virologists at the vet school, they actually started their PCR testing on live patients. They're the first lab in Louisiana to do PCR testing in the state. Again, there was two virologists. They got it all set up, and they realized they had to have a medical director and had to run it through an accredited laboratory, a CLIA accredited laboratory. So they partnered with a pathologist in Baton Rouge, a large group of pathologists called PGL or Pathology, a group of Louisiana. On March 22nd, we were sent our kits. Abbott started sending kits to Ochsner and LSU Shreveport. They had Abbott kits on the 23rd. Actually, on the 21st, I was told that the only people that would be running COVID tests in Louisiana would be these research labs, the ones at Tulane and the one up at Shreveport, and the one at River Road in Baton Rouge. So on March 25th, Tulane Medical Center started testing for Tulane patients and University Medical Center patients. LSU Shreveport started testing through their emerging viral threat laboratory on March 28th. The Tulane researchers started testing on April 1st. By mid-April, most of the medium-sized hospitals had other platforms for doing COVID testing by other commercial instruments like Panther, the Abbott ID Now, Cepheid, and BioFire. In June, Delta Pathology, a large group of pathologists here, opened up a separate lab. They had been running hospital procedures such as on the Cepheid and BioFire in some of their hospitals. They opened up a larger lab, automated PCR lab, and I'll talk about it in just a second. But these were all the major players in the state starting in March and April. After we started testing, we still had problems. There was shortages of viral transport media, swabs. We took transport media and split it up. That comes to 3 mils to a tube. We split it up to to 3 tubes with 1 ml each. We ran out of swabs. The PhDs in their lab, they started making swabs. We had to validate those swabs. We had problems getting reagents from Roche. They would promise us 2 weeks at a time, and then they'd reassess whether or not we needed them, depending on the number of cases of COVID in the region. Getting timely results to clinician, we found was a problem. If you didn't have the computers to transmit them electronically, certainly an impact on the turnaround time, sometimes exceeding 4 or 5 days for some of the labs. The development of the rapid test, particularly the Abbott ID Now, it was considered a point of care procedure, and once the clinic administrators and hospital administrators found out about that, we had quite a few of those flying around, and just about anybody that someone in administration thought could run them would be running them. But most pathologists did not feel like it was a good procedure to be tested as point of care at that time. All of these tests were and still are under emergency use authorization, and this has actually allowed these companies to change their protocol. Actually, in the last month, Abbott, with their ID Now, sent out some new policies. They used to say that you could run the specimen after up to 2 hours after obtaining the specimen. Now you can't run the specimen after 1 hour. Once it's obtained, it has to be run within an hour. And they also changed the policy that you could run it on any patient. Now they're saying you have to run it only on symptomatic patients. You don't have valid results on asymptomatic. The other problem that we saw is that the instruments we were now using to test COVID, we had been using them to test other procedures, other laboratory tests for other diseases. We were no longer able to do some of those procedures. This is just a list of the labs and their platforms, and the total number of tests. I'm not going to go down in detail, but you'll notice most of them have multiple platforms, which is important. The last one, Delta Pathology, they opened the lab up in the end of June. They're capable of doing 2,000 tests per day now, but the instrument they have, the Thermo Fisher, the automated PCR, they could ramp up to 20,000 to 30,000 tests per day. Right now, in Louisiana, these major labs are producing about 10,000 tests per day, and we've done a total of 2.75 million tests in the state on state citizens since the COVID pandemic began. Back in March, April, and May, Louisiana, not March, but April, May, and early June, Louisiana was number 1 and number 2 in the country as far as the number of COVID tests performed per capita. And I think most of the people in Louisiana, at least the pathology organizations involved with the COVID testing, were proud of that. So the lessons learned, start developing testing immediately. You're going to know about pandemics. We knew about this back in December. Of course, we didn't know anything about the organism, so we couldn't really develop PCR testing until we got the genome on it. But as soon as you can, you find team players. You don't want naysayers. There's always going to be naysayers in emergencies that say things can't be done. You want people that run towards fires, not away from the fire. Homebrew testing, that's the type of testing these PhDs were setting up. I use the word homebrew as a matter of respect, because these PhDs pretty much developed these from scratch, along with some help from the CDC. It's also very important that you have multiple platforms if necessary available to you, multiple different methods, different companies available, because you're going to run out of reagents, you're going to run out of supplies, instruments are going to go down. You want to be able to switch over to a different platform if one platform is not working for whatever reason. You need to be able to predict the problems that are going to potentially occur and be creative with their solutions. Like we split up our media transport media into 3 vials. We created swabs. We had to validate them to make sure they work. You need to have the infrastructure necessary for reporting the results. And you need to look at this, that you're helping each other. The competition here is the disease and the patient. It's not another hospital. It's not another lab. You have to help each other. And you need to, if you have to reach out and help another lab. If they need your help from you, you should offer it to them. So those are the lessons that we've learned so far, and we're still very early in knowing everything we need to know about this pandemic, and I'm sure we're going to learn more lessons in the next few months. Thank you.
Thank you, Dr. Crawford. So if you could stop your sharing, I will turn it over to Dr. Rohde. Thank you. Yeah, and Dr. Love was actually our presenter last month, so that's a nice connection to tech grads who really were instrumental in helping testing here in the state. All right, Dr. Rohde, you have capabilities now.
All right. First of all, I wanted to thank Dr. Crawford. I really appreciated his talk. Just for some context for the audience, I was at a rabies international diagnostics meeting last week, international in all the rabies testing in the Americas. There have been more EUA tests developed for COVID in the past year than for rabies diagnostics in the past 15 years. So it just shows you the effort. I think that, you know, one of the good things of this pandemic that's come along is the tearing down some of that red tape and moving forward on some of these really important pandemic issues. So I'm going to hop over some of these slides just to get to what I wanted to talk about. As everyone knows, this thing kicked off, we think, in November, December of 19, and then we moved into this February, March timeframe. And what I'm going to do in my talk today, a little bit different than Dr. Crawford, I have been involved in a lot of the testing issues in Texas. My background is public health. I worked for the Department of Health for 10 years and for CDC as a visiting scientist for a couple different stints, but I've been in academia for the past 18 years as the director of the Medical Lab Science program. So I just want to do a quick update. I know all of you may know some of this, but it's going to lay the framework for what I want to talk about, which is primarily how Texas State University dealt with this. So we're going to come at the angle of an academic setting. So if you didn't know, this is up through last week, 214 countries now involved, over 45 million cases. I think we're pushing 1.2 million deaths now as of last Friday. Many of you know that the United States leads the world in deaths as well as cases. We're responsible for about 20% of the cases worldwide, and you can see some of the other major players here, including India and Brazil and others. When you look at the top 10 countries with respect to this is from the Worldometer website. If you're not familiar with that, it's a pretty nice place to check out for information. The USA is leading that with respect to cases and otherwise, and you can see the top 10 as you go down through that number. It's always important, obviously, to think about perspective with the different populations of the country as well as mortality per 100,000 people and so forth. If you look at the United States, unfortunately, Texas jumped back to #1 last week with respect to cases and so forth. And you can kind of walk through that top 10. I should have jumped and looked at where Louisiana sits. I didn't think about that this morning when I was putting this together, but those are the top 10 states as of last Friday. And so here's where I kind of want to jump in and start talking a little bit about Texas State. So Texas State University is a university that has roughly 38,000 undergraduate and graduate students combined. We have about anywhere from 1,500 to a couple thousand faculty and staff at any given time. And like most universities, we were minding our own business and doing our own thing last spring and moving along, but watching this outbreak and pandemic as it unfolded across the country. And we went to a fully online virtual learning mode on Friday, the ominous Friday, March 13th. It's also a day I mentioned to the sponsors were my son, who was doing a study abroad at the London School of Economics, got yanked home. So this really impacted me both at a professional and a personal level. We stayed online throughout that spring. It was very challenging as a medical laboratory science program director to think about transitioning to virtual clinicals. Fortunately, we were able to complete about 3 out of 5 of our clinicals, and the other 2 we had to utilize virtual online learning as best as we could, as well as doing some things in-house. That status remained at Texas State University through the summer of 2020. And then this past fall, we actually came back face-to-face. However, I will state that there are many accommodations. I'm going to talk about that in the next few slides, but in reality, most of the healthcare professions like medical lab science and nursing and physical therapy and so forth, and in our College of Health Professions came back because we basically needed to get back to hands-on laboratories, hands-on practicums and things like that, but all with respect to safety initiatives that I'm going to talk about. Many of the other programs, especially at the freshman and sophomore level with the large classes with liberal arts and other things like that, have primarily been online. So it's not a full campus that's back, but it's certainly quite a few. This is just to show you our dashboard a little bit. It's called the Roadmap to Return, and there's the link at the bottom if you're interested in looking at this. So due to my background, as well as my expertise as being a clinical virologist and microbiologist, I was tapped from multiple work groups, and you can kind of see those here to the right. The 2 primary work groups that I spent much time on were health, wellness, and safety and continuity of research, but you can see there were primarily 8 of these with over 120 different university leaders involved. So this was a massive undertaking for a university of our size. I can't even begin to tell you the number of hours many of us put in putting together this roadmap as we moved forward into the fall, especially. So just a snapshot of some of the things that we did. All Texas State students that were coming back in the fall had a mandatory health and safety video that they had to arrive. Basically, they had to quarantine, or they were asked to quarantine for 14 days prior to the start of the fall semester. We have a universal face covering mandate at Texas State University, both inside and outside of the university. Of course, we sometimes realize that this is difficult when students leave the campus because they do have their own lives to lead, and we can't govern them or police them once they're off campus, but we do have that mandate here on the campus at Texas State. We have a variety of health and safety measures, including physical distancing, face coverings. We have hand hygiene stations all over this campus. We implemented daily and weekly cleaning and disinfecting of most teaching and common spaces, as well as looking into ventilation. If you weren't aware, most healthcare systems and elsewhere look at about 12 air turnovers per area. It depends on the square footage, and so we tried to look into that with many of our teaching spaces. And just for example, in my program, we have 20 students per cohort. So I have 20 juniors and 20 senior clinical lab science students at any given time, and they're separated in different classes. But we were able to find a room in our building that seats 80 people. And so we're able to do those lectures still in person by spreading out in that room. And there's probably 10 or more feet between each student. They're all wearing masks and all following the typical hand hygiene and things like that. We have pushed this out through different messaging, including social media and websites and emails. The students and even faculty and staff took a Bobcat pledge. We're calling it the Texas State Respect. It's actually a hashtag, TX State Respect. People are asked to do daily assessments and monitor their health. And if you are identified as testing positive for COVID-19, we have an anonymous portal called Bobcat Trace. that allows you to input information about your test status, and then that can be followed up with a contact tracing feature. And so if they're tested, we ask, and they're positive, we ask for them to stay in isolation until they receive those test results. Here's some information about our kind of epidemiology of the university. Our goals basically were obviously to reduce the risk of COVID-19 and transmission, to create a learning environment that we felt was safe. We are just past the mid-semester effort of 8 weeks. And what we've seen, and we expected right when students came back to campus in late August, we did have an increase in cases. That was expected, especially around that Labor Day holiday and when they came in from being home all summer. After the 4th week, we had about 115 positive cases that we were aware of and had tested for and people reported. That was the highest number in a week at that time. Last week, which was the 8th week, the midterm of our university, we were down to about 30 positive cases being tracked. And so the number of cases actually were declining despite increased testing over the course of the semester. It's increased from about 500 tests per week to up to 1,200 tests per week. And we keep track of that on our dashboard. If you didn't know, the positivity rate, when it's below 5%, that's kind of the benchmark that we believe is an indication of sufficient testing providing a low transmission in the community. So when we had low testing early in the fall, we had our highest rate showing around 9.6%. But with the addition of free testing on campus and an increase in testing, those rates in the last 3 weeks have dropped around the 1 to 2% range in our community. So we believe that testing is essential. We're not testing everybody, but we are starting to do random sampling of faculty, staff, and students to kind of get a better feel for the prevalence and incidence on campus. And testing is free on campus. We have a kiosk. We have a portal that students can enter and make an appointment and go get that done, and the test turnaround is about 24 to 48 hours. The Student Health Center, we have 18 contact tracers that have been trained through the Johns Hopkins contact tracing program, and so they do most of that tracing for us. And we believe that this has worked quite well. We have not seen any evidence of transmission in classrooms or residence halls, except for a few cases where we documented roommate transmission, where there was a couple of people in the room, and then we followed up with that. We do think that social gathering with friends or family continues to be the primary source. So off campus, in apartments, going home, having parties, whatever students are doing once they're off campus. And so that's been our ongoing efforts in that area, and we feel pretty good about it. Here is our quick look at our dashboard. This is again at the end of last week. I just took a snapshot. We do this weekly. So we have about 25 cases as of 10/28. This shows you the number of student cases cumulative since March 1st, about 732. We've had 48 faculty staff, about 780 total. Hayes County and Williamson County are counties that the university, the primary Texas State University campus is Hayes. Williamson County is where we have some programs up in Round Rock, Texas, which is north of Austin, Texas. So it just gives you a nice week-to-week look at what's going on in the area. You also see this on when you look at our dashboard, so you can see it as a weekly shot. We are kind of in that range of about 1 to 1.5%. We haven't seen, I haven't seen last week's updates yet, but I think it's in that 2% range as it's been for the last month or so. One of the things I was pulled into just as part of this conversation I wanted to share, because it's so important, right, in science communication to get people on board with this with respect to messaging and getting people in the public as well as the university community to understand the science. I think we all know how critical that is. Struggling with that as a country. I was pulled in to actually help create some videos. I'm not going to show this right now, but here is a link if you're interested in checking it out. I basically used, and you've seen this all over the country in different ways, but I went into the lab with some students. We tested various mask coverings, and we basically utilized students laughing, talking, speaking into blood agar plates. And we know that they're bigger than viruses, and we know that it's not, the perfect experiment, but we wanted to show students and faculty how masks can work. They're not 100% effective when it's not an N95 mask, but they do reduce transmission of respiratory droplets. It does drive down the distance that those particles can travel. And you see it in other countries in epidemiologic studies of how that's working. So just trying to get that Texas state respect, that messaging out there with respect to, protecting those around you. And we're really working at that. It's been ongoing. We're fighting right now a little bit of that complacency and pandemic fatigue that you see around the country. I just had that conversation pre-Halloween last week with my students. And, honestly, I told them, do you want to not finish the semester? I mean, your professionalism in health professions is protecting others. So, using some of that type of peer pressure and pressure to kind of get people to try to follow that. Just as I kind of end up here, I don't know if you guys saw this Time magazine publication last week, but I thought this was very timely, no pun intended. I mean, just literally before a presidential election. Great timing, by the way, for this webinar. Congratulate you on picking today, but look at these different surges from the first wave, and you can kind of see now we're entering this danger area, moving into late October and November. And we've actually also set a new record. You probably heard this week of pushing up to 100,000 cases per day now. And so again, that's a message if you're dealing with this on your campus that you can continually talk about. Again, realizing people are getting tired of hearing it, but something I continually speak about, that the virus really doesn't care. The viruses never get tired. We get tired, right? And so they will continue to move through the system. And much of what Dr. Crawford mentioned, just some take-home messages, and this is just a very brief overview of Texas State's efforts, but we do feel that testing and contact tracing works. As a public health professional, I hope and pray that a national strategy gets implemented at some point. There is no reason that we can't work harder to make this happen if we can do it together. Distancing works, mask work, hygiene of your hands always works. Crowding is critically important to be careful, especially in ventilated areas. There's numerous studies now out through CDC, WHO, and private researchers showing that monitoring your health is important. My wife is a pre-K teacher, and she's always like, this is common sense. I mean, we don't even want you coming to school if you have the common cold or the flu, and yet we've all done that, and we know it. But certainly it's something to kind of be on top of now with this virus with no vaccine. Being careful with where you travel, watching out for that pandemic fatigue, and then just being a leader and modeling these safe public health behaviors. Because if we don't do it, who will, right? So we need to really try to think about that. And I'm going to stop here. I know we don't have time to talk about this right here. This was from another talk I did just about some lessons learned. Maybe we'll get to some of this information later in the conversation around the question time.
Yeah, great. Thank you, Dr. Rohde.
You bet.
Yeah, if you stop share, I'll get Dr. Roy on here. Yeah, and on your testing stuff, so Louisiana Tech, well, Ruston has had the National Guard here for several weeks now. I know because I go every week to get tested, because my children go to school and that's an exposure, and so they've been here doing free testing for 6 or 7 weeks now. And they're here this week for those of you on here who are students. It's worth going and doing just to keep everybody safe. So there's my plug for testing. Dr. Roy, it's your turn.
Can you hear me, Jamie? Great. So again, thank you, Jamie. Thank you, Mary, and everybody else for inviting me to this. I'm going to switch gear and really talk about the other side of this equation, which is vaccines, when vaccines are coming, what are being developed, and what about therapeutics. But before I do so, just to wrap up on the diagnostics test and echoing what Byron and Rodney just shared, is really testing works and masking works. This is Georgia Tech, very much the same story. Remember, we are in the middle of Atlanta, midtown Atlanta, in the state of Georgia, which has a positivity rate of about 10% overall. This is homegrown, homebrew test developed by a bunch of engineers and biologists. We don't have a medical school. We don't have a nursing school. We don't have a public health school. It's a primarily tech school. And this was done, it tests about 1,500 to 2,500 people a day. We have done over 100,000 tests. It's a saliva-based test. And since early September, we are below 1%, and we have not gone above 1% in campus. And this, to me, is a testament to what the previous two speakers just alluded to, that this works. So I just wanted to throw that out, that in a non-medical, in an entirely tech campus, this is also very much possible to hold down the virus transmission. So I want to really talk about vaccines and really think about how we are approaching vaccines for COVID and what's going on globally, but also the tremendous cooperation and the tremendous amount of really collaboration that is happening, both from the public sector, the private sector, the government, the FDA, the manufacturers, and so on. So I really encourage everyone to read this article from about a month back, a little more than a month back, from Florian Kramer, a very, very respected immunologist, who wrote in Nature a very short review. And these are three figures that I picked up from that review. On top, you see our traditional vaccine development pipeline. You start with, think about flu vaccine, think about mumps, measles, rubella, whatever that is. You go through several years of preclinical studies, process development, toxicological studies. You think you have a good candidate. You ask the FDA at that point for an investigational new drug or an IND. Then you go through a phase 1 trial, a phase 2 trial, a phase 3 trial, which takes about 5 to 7 years. You then submit, if everything is positive, safe and efficacious, you submit a broad licensing or a biological licensing agreement or a BLA. And then they go through an year or two of regulatory review, either through the FDA or EMA is the European agency. And then you create this large-scale production and distribution logistics. So about, earliest 10 years, more like 15 years or longer to develop one specific vaccines that we all get. What we have been trying to do in COVID is shrink that timeline to 10 months to a year and a half. Remember, we started somewhere around February to make this. Maybe folks in China started slightly early. But we have been trying to squeeze 15 years worth of work in this 10 months to a year time frame. So bear with the scientists and regulators that we are not there yet, because two things that needs to happen. We need to prove that it's safe in a mass scale. We need to be able to manufacture that to treat, 300 million people. And we need to make sure that it actually protects. And those are neither of them are that easy to happen. So we are really squeezing this, and with regulatory insight and regulatory, I would say, innovation, how we can, at least if not a full approval, get what's called an emergency use authorization, maybe by the end of this year, in the next couple months or early next year. But what I would tell you that even though the FDA approves or provides an emergency authorization even tomorrow, the capacity of manufacturing them for hundreds of millions of people does not exist in any companies. So it will take months, maybe 6 months, before everybody who could get a virus can actually have a vaccine available for them. So that's an issue we can go into during the Q&A, because that's one of the most critical understanding for engineers and technologists. How do you create manufacturing infrastructures for this? If I can get to this. So this is sort of the second slide, the various types of vaccines that people are working in. On the left top is kind of the cartoonish diagram of the COVID virus. It has it's an RNA virus, so it has an RNA genome. Then it has an envelope protein, and then it has the spikes. And these spikes is what it uses to get into our cells using a receptor called ACE2. And then various people are trying different strategies. So most of the vaccines that we all get these days are either these live attenuated vaccines or killed viruses or pathogens. And this is sort of the workhorse of vaccines today. A lot of the work has been focusing on creating vaccines against the spike protein, which is these orange spikes in the cartoon, to make antibodies against it so it gets blocked and the virus cannot enter the cell anymore and gets recognized by the immune system and gets killed. A lot of the vaccines are for protein-based. Some are using what are called virus-like particles. Some are using inactivated viral vectors like adenoviruses and AAVs like the Oxford AstraZeneca vaccine, and so on and so forth. There is a DNA vaccine that's happening. The Moderna one that you keep on hearing a lot is an RNA vaccine, is using an mRNA that encodes for the spike protein. So your own cells produce the protein and that acts as the virus antigen, and so on and so forth. So many, many strategies are being involved. And this is an interesting summary of where these vaccines are. Obviously, most, 90% of them, 80% of them are in preclinical phases. So they're in this animal mice study phase. There are a good group that are in phase 1, just testing safety. Some has moved on to 1, 2, and then phase 3 is quite a few in different countries. Here, the vaccine from Pfizer, the vaccine from AstraZeneca, the vaccine from Moderna are in phase 3 trials now. There are 2 that are nominally approved. These are really the one that is approved for the Chinese military and the Russian government. So those, you can take with a lot of grains of salt, whether they're what they are, we don't really know, and what their profiles are, we don't really know. So we are holding our breath here about some of those working out, some of these phase 3 ones hopefully working out. They're primarily RNA vaccines and some inactivated vaccines of the virus itself, some recombinant spikes protein, which is the protein itself, and most of them are vector-based vaccines, which is using adenovirus or adeno-associated virus type, other virus carriers to make these vaccines. Remember, though, one thing. In the history of humanity, we have never had a vaccine that are in these greens and blues. We have never had an RNA vaccine that has been approved for use. We have never had a DNA vaccine. We have never had an adenovirus vaccine that has been approved. These are still the top candidates right now. So there is a lot of thinking in the FDA and the regulatory space to make sure that we are safe. And we will get there. I have full confidence on the FDA that they're not going to rush through this without looking through. Once they say that this works, I will take the vaccine and I will give it to my kid. If it's approved for kids, I will give it to my wife if it's approved for all populations. I know the folks at the FDA who goes through this. They're incredibly professional people. And I know some of them very personally. And I know that they will do a great job in thinking through this. some of the work that's going on in our lab, and I'll quickly end with a couple slides here. In July, so my lab works at the interface of materials and immunology. And we have had a long-standing grants from NIAID, which is the National Institute for Allergy and Infectious Diseases, on looking at vaccines and improving vaccines. So when they started, we pivoted in the lab, we wrote a short proposal, and they provided us with some funds to work on COVID vaccines. And the goal is not really to compete with the ones that are in phase 1, phase 2, phase 3 trials or in companies, but to really think what would be the next generation of vaccines and how we can improve the responses by fully knowing, not knowing really what kind of vaccines we are going to get. Remember, our ability in history to create vaccines against a pulmonary virus, against an inhaled virus, has been very, very poor. We do not really have a strong vaccine candidate for flu that lasts long. Absolutely get the flu vaccine. It works, but you have to take it every year. RSV and other respiratory disease, we haven't really had a good vaccine candidates for that. So we have to think, what's the durability of the vaccine? How long it's going to last? 6 months, 3 months, a year, 2 years? So there are a lot of things that still needs to be researched. So our approach has been to really think about adjuvants, which are molecules that people look at in the vaccine research world that are derived from pathogens. And there could be RNA, there could be DNA, there could be various lipids, which stimulates our immune response. And people give this together with the vaccine. With the protein antigen, with the virus, with others, to increase our immune response. And these are called pathogen-associated molecular patterns. So our lab works in various types of adjuvants or camps. And we have been, since the last few months, evaluating how various combinations of these adjuvants can affect the immune response against the spike protein of SARS-CoV-2. And eventually, once we get through the protein data, we will get into how to improve the mRNA vaccines as well. So this is still ongoing work. We have a team of 4 or 5 people working on that. I want to end with this one, but I want to talk about therapeutics, because it is absolutely critical to understand that vaccine is not going to be the answer for us to be all clear. First of all, as I said, it will take months and months for us to get to the capability to be able to have enough doses for everybody. If you're interested, you can look at a National Academy of Medicine study that came out last month as to the hierarchy of people who should get vaccinated in which order, okay? Healthcare workers first, and so on and so forth. So it's going to take a while for people to get vaccinated. We know, even with our tremendous SciComm efforts and public health efforts, about 30% of the US population will likely not take a vaccine by choice. You are not going to mandate it. These people will get sick. Even with a vaccine, between the time you get vaccinated and your immune response kicked in, that's about a week to 2 weeks. You still can get infected. For this large group of people, we need therapies. Unfortunately, most of the therapies we have been trying for the last 7-8 months are not showing very good results. Remdesivir is now approved by the FDA, but if you look at the data, in one study, it showed some effect. In most other studies, it's okay? So it's not doing very well. It's an antiviral. There are many other antivirals out there. Regeneron and Eli Lilly's monoclonal antibodies. This is what the president got, the Regeneron one. Eli Lilly is what Chris Christie got. But both these two has just this past week come out with data that they do not work in patients who need oxygen support and are hospitalized, which means you can only use them, and they have stopped their trial for those patients. You can only use them in early cases after diagnosis in an outpatient basis. A third type of, these two were antibodies that block the virus. This last one, tocilizumab, is an antibody that's used in arthritis, that's used in cancer patients quite a bit to prevent cytokine storm. It's an interleukin-6 antibody Roche. There was a lot of hope against this because we know that severe SARS-CoV-2 patients have high levels of interleukin-6 in their blood. But that study in a couple of weeks ago also failed, did not work, showed no improvement. Interferon therapy given early on has shown some promise. The most promising so far has been the cheapest drugs, dexamethasone and steroids, which are broad-acting immune modulators. But you have to time it right. And again, the efficacy is fairly modest. And there are cell therapies like MSCs and NK cells that are being tried also, which could show some promise. So this field is absolutely critically needed. And I hope a lot of you are going to work on this because even though we develop the best vaccine, we would need this to save people from dying. And so with that, I'm going to stop here and hopefully we'll have a lot of questions as we go along.
Yeah, thank you, everyone. I thought that was maybe even better than Dr. Calver and I anticipated as far as like everybody complementing each other really well to talk about how tests are being done, the importance of testing and other strategies, because we can't rely on vaccines and treatments right now. We have to each do our own part to keep each other safe. So that goes, I think, to our first question maybe, and please feel free to type in your question. So there's a question, if there's any advice for students on how to avoid bringing home COVID at Thanksgiving. 14-day quarantines are not always possible. Is there a testing or quarantine strategy that can help? So as you all know, because you're at universities, our students are about to leave, see their families, and then they will return to us for a few weeks. So aside from masks, avoiding social gatherings, testing, are there any other thoughts?
You know, it's a great question. It's interesting, you know, again, thinking about strategies, one of the frustrating things for me, and maybe the others can chime in on this, is that there's no rhyme or reason of what universities are doing. So at Texas State, we're coming back. University of Texas, Texas A&M up the road, down the road, they're not. They're going to stay online. My son's at Trinity University in San Antonio, they're not coming back. I kind of like that idea personally. I'm not speaking for the university here, but personally, it kind of makes sense that, you know, you just stay home and then think about the spring semester. But if you are going home and you are coming back, beyond all the things you just talked about, I think people really need to be careful with respect to, you know, gatherings, maybe keeping it to just the immediate family if you are going to get together. And even then, I'm even talking to family about, you know, eat outside if the weather's nice, get into some well-ventilated areas, wear a mask. I mean, there's nothing wrong with wearing one even when you're around each other. I know that's going to be challenging, even personally. So I don't know if there's a best answer. I just hope people are careful to monitor their symptoms and kind of think about others as they're coming back to campus. Challenging.
Yeah.
And do get tested right before
Right.
and right after. Tested before. If you are positive, do not go, and save your family from that ordeal. And I know it's hard, but got to do it.
Yeah, I think some of that pandemic fatigue and testing fatigue is if I don't have symptoms, I don't need to get tested. But unfortunately, that's not really the case, right? So, or I don't want to find out because then I have to stay home or I can't go home or whatever the situation is. And it's so important to know.
But think
your
of
health
it
status.
this way, that you're not doing it just for yourself. You got to do it for your family. You got to do it for your friends. You got to do it for the community, and that's the thinking of service here.
Right.
You know, what Dr. Roy says, it's so important and it's so simple, isn't it? It's such a simple statement when he says that. I think the frustration that our colleagues and I keep dealing with are those that just don't see it that way. And, you know, that's a whole nother conversation, but we have to figure this out with respect to treating this like it's a wartime effort. You know, it's an invisible enemy. And we would be doing all sorts of things if this was a terrorist attack or anything else. And it is an attack on our country, albeit from a different agent. So that's kind of been the angle I've been talking with people about, is trying to protect my mother, who was just diagnosed with cancer during this pandemic, and my students who are graduating and working on the front lines. You know, if we run out of healthcare professionals that have spent 20 years of their life becoming specialists as intensivists, we don't replace that in a year. You know, we could wipe out the expertise in ways we've never experienced.
So we have another question for you guys. At the beginning of COVID-19 pandemic, you know, statements were made about children being the carriers. Is this still considered a myth or a fact?
Nobody wants to answer that.
I was going to let Dr. Crawford or Dr. Royce start, since I've spoken twice. But
Dr. Crawford, do you want to start?
I'll be glad to. I think it's a myth that they're carriers. However, it seems like the younger people, especially younger kids, they don't realize the gravity of the situation, and they're less likely to practice social distancing. So they may have more likely to become infected, but I don't think they're just carriers running around trying to infect us all.
So I think if I understand the question correctly, I do think that there is enough data to show that children can get it. Children do not, especially younger children, get as sick. Now, absolutely younger children are getting sick. A small percentage are getting what's called the MISC-C, which is a multi-immunomodulatory organ failure disease, and you don't want your kids to get that. But the bigger question is, children can transmit that to adults, even if they are asymptomatic and they're sick. Are they hugely transmitting? I think the recent most data that I have seen says that younger children are less likely to transmit than teenagers and older children. High school kids are almost as likely as adults to transmit. So I think there is still tremendous need for hygiene, distancing, masking, even for kids. That is, at least in my opinion. I know Rodney, as a public health professional, you would know better.
yeah, I echo your sentiments exactly. I did see a recent study. It's kind of interesting. CDC put out something the other day about looking at, now this is one study, so again, take that into context, but it was something along the lines that they're seeing less infections in that age group, like elementary school and lower, as opposed to high school, college, and young adults. So I do think the evidence is starting to show that they can certainly get the disease, as he mentioned, and that multi-inflammatory syndrome is something you don't want to see. So it's still important to wear masks and be careful. But it does beg the question, in a year or two, as data collects, will we go to a format where maybe elementary schools and lower kind of rethink how they're doing things? Maybe we keep high schools and colleges more online. Maybe we're able to do more with younger people. My wife's a pre-K teacher. She has a classroom of 16 4-year-olds. And you can imagine they didn't start back until October. So she's just now back. So I'm worried. I'm worried about her. But she's masked up. All the staff and faculty are masked up. But you can imagine policing 4-year-olds. They're all over her. You know, they want to love on you at that age. They want to sit in your lap. They need comfort and things like that. So it's a challenge. It is a big challenge at that pre-K, early school age.
And you know, our school district has both a face-to-face and a remote option, and my daughter's home. She is not going face-to-face, and she doesn't like it, but
yeah,
she's home.
Yeah.
So given some of what we talked about and some of what's in the news right now, I'm going to ask this question that came in. There are a lot of questions, so thank you everybody for writing in questions. What is the policy for case reporting? I have heard medical offices that are not required to report COVID cases and others that do. Who determines these reporting policies and what are they? So I don't know if I need to feel comfortable answering that. I thought I'd put it out there.
That sounds like a Byron question.
And my understanding as a physician that all infectious diseases, at least in Louisiana, are supposed to be reported. And I'm not aware, I mean, I've known COVID patients that tell me that the State Board of Health has contacted them about it and asked for their contacts, but I never hear that their contacts are ever located and spoken to.
Right.
But in Louisiana, there is a state law that all infectious disease are supposed to be reported. Physicians' offices are supposed to report them, and hospitals. I don't know about schools.
Yeah, I know one of the issues, I think I would back that question up into the whole strategy around reporting and testing. It's kind of frustrating, I think. I kind of feel the frustration in the question that was asked. Is there a clear understanding, even from state to state, how that's conducted? I know in Texas, we have notifiable diseases that physicians and offices are required to report. And I'm assuming all COVID cases are being reported. But I think I feel the frustration sometimes when people ask that, and they're kind of, wondering, why do I keep hearing these data, being, shifted in these different ways? So I think that needs to be cleared up at the national level, with states following suit on algorithm, exactly what we're doing and what's being reported and so forth. Not an easy thing to do. I'm talking long-term lessons here again. This is stuff I think we need to figure out, because people want to know, is the data trustworthy? Is the reporting trustworthy? Again, I'm not a physician, so I defer to Dr. Crawford on this, but it just seems like that's part of the public-facing issue, is some of those types of questions that are popping up.
Yeah, I have another issue with the reporting. What do organizations, even our medical school, we had someone in our department that developed COVID a few weeks ago, and the policy is that you don't tell anybody. Nobody else in the department is told. Usually leaks out. But I've seen that with other people in their work. The organizations say it's a violation of privacy to tell everybody else on the floor where that person worked. So that's something that I don't think anyone has really tackled and developed an answer to. How to handle the people that they are exposing at work.
Yeah, for us, we cannot tell anyone. I mean, we can tell very vaguely to a very small community who are working with this person, but it's up to the university health office to do the contact tracing. How much of that gets really done very well, it depends upon the university and their health offices. But yeah, locally we can't share because of privacy reasons. But yeah, hopefully the university health offices are.
Yeah, that's what Texas State's the same way. The HIPAA concerns, and then, they very broadly will report, we have identified a person in this dormitory on this floor, so you may want to consider staying home, those types of reports. Difficult questions.
Go ahead, Mary, you have a question?
Sure. So if testing is widely available, how often would you recommend people and students that are attending in-person classes or who are involved in getting out and not completely quarantined to get tested?
Rodney, you want to take that?
So the first part was how often to get tested
Yeah, so
if you're a student?
Well, if you're out and about, if you're interacting, even where you're wearing a mask, how often should you be getting tested?
Well, I mean, there's my personal answer on this, and then there's the organizational answer. I mean, first of all, personally, I don't think someone should be, testing every day. I think that's going to be a problem with resources, both in personnel and in products. Of course, if they're healthcare professionals, that's kind of a different answer, because they may need it, every several days or something like that if it's work related. But in general, if you're interacting and you don't have any symptoms and you're kind of monitoring what you're doing, I actually don't believe you should be tested every day or every 2 or 3 days. Maybe if you have a reason around it, for example, you're going to travel home or you're going to travel to an area where you may have immunocompromised individuals, then certainly that might be a reason. That's my own personal answer to that. Again, thinking about resources and trying to be careful in how we're using laboratory supplies. I mean, we're still seeing issues with supply chain issues on pre-analytic RNA isolation and things like that for certain tests. So I'm a little more conservative on just testing everybody in a university. I don't think that's quite the answer. More of a stratified approach as to the risk and why you need it. Did I sound like a politician there or what?
So for us, the recommendation is once a week. And they have asked us to, you go Thursday, you go every Thursday. Put it in your calendar that X day, every week, I'll go throw my spit into a cup and I'm done with, right? And we have a multiplexed testing process in-house. But that supply chain, if anything we have learned from this debacle is how ill prepared our supply chain has been, from masks to face coverings to RNA isolation kits. We do vaccine research in the lab, and I can't get RNA isolation kits because everything is going to the testing lab.
Yeah.
Right?
Yeah.
So it's a very difficult supply chain issue. So don't overburden it. But if you're going to classes, if you're going into social gatherings, my recommendation is once a week is what our university has gone into, and that kind of has worked because. And the other thing is, I like the stratified idea because 90+% of our cases are undergraduates, and a large cohort of that are from Greek housing. Believe it or not, it is what it is. And so we have targeted that by placing testing stations right outside of the Greek housings and bringing the testing to them and to the dorms. And that's our primary stratification. We are not testing faculty,
right,
Austin or others who are.
Yeah, that's exactly what I was talking about, because we're doing the same thing with our athletics, with our child care center. So we're worried about that group. So yeah, that's exactly the kind of thing I was thinking about. Do you worry about, Dr. Roy or Dr. Crawford, do you worry about explaining the test results with respect, some test is 80, 70, 80% sensitive, or do you just, we're just going to go with it and trust it, or?
So what we do is our basic is an in-house PCR surveillance test. And if that comes positive, we do a confirmatory test after that. So it's only twice tested positive is considered part of it.
So I know that Dr. Rohde has to go soon. So I just guess one of the things Dr. Calderon and I really like to ask is what we've learned from this and like where do we go? We've had a great discussion about that, but I guess we could just start with Dr. Rohde and any last sort of.
Could I
oh yeah, please, Dr. Crawford.
Can I comment on the last question?
Oh yes,
please,
yes.
Please.
As a physician, I feel like you should only be tested if you're symptomatic. I see people all the time who kind of call me up, say they think they've been exposed and they want to be tested. I say, well, the test is only good for that moment. It could be negative that moment, and then tomorrow afternoon it would be positive. So I think once we become symptomatic, we've been exposed. I tell them wait 4 or 5 days, at least 3 or 4. If you've got it, gives the chance to replicate more and be more likely to turn up positive. But they want to know right then. The other thing is that I don't know. Krish said that y'all use a saliva test, which is PCR based. I don't know what Texas State University is using. Is it?
We're using the Abbott ID test for the screen,
Abbott. Okay.
And then we use the Cobas Roche PCR test for the confirmatory. We send it out to a reference lab.
All right. Well, those are all good tests. I don't know if y'all are familiar preparing for this. I've got across that Abbott has a new system for its antigen testing for byproducts of infection. It's a system called Navica. They said it was coming out in September, where they would be able to do 5 million tests in a month, and
Wow.
It would expand in October to 50 million tests a month that they could do. And basically, it's a little card that you take, like a credit card. You have an app on your phone. You go get tested. It's an antigen test. It takes about 15 minutes. Costs $5. You get results in 15 minutes. The fact that they said that they were going to be able to do 50 million tests in October, I couldn't find anyone that's using the system. They must be having problems with it, but they are definitely working on that. And it's basically for large organizations, so big businesses and schools, that they can screen on a regular basis. So it's something to keep an eye out on coming down the tubes. There's always the capitalism here in any crisis. And at $5 a test, at 250, at 50 million a month, it's $250 million. So there's a lot of incentive for them to get that out.
Yeah, I think those are all good perspectives. So yeah, Dr. Rohde, do you want to give us the 2 parting words as we wrap up?
Sure. So I think you mentioned kind of just some parting thoughts on lessons learned, things like that.
Yeah, sure.
So,
and like where people can be involved in the future. We know
Sure,
too much of a
sure.
chance for that, but
So there's so much we've learned and so much we still need to learn. So let me just try to hit a couple of points before I have to go right around 5. I think one thing I would like to kind of point out to the audience and to those that might listen to this in the future is just how desperately we need future healthcare professionals. In the medical laboratory science realm, whether you're an associate, bachelor's, master's, or now doctoral level, DCLS is practicing, there's about a 46% vacancy rate across the United States. It's actually worse than nursing, but people don't always see us. We're kind of behind that laboratory door in the hospital and public healthcare setting. People are retiring left and right, just like in any other profession. And so just keep that in mind and share it with people down in the levels of junior high and high school. It takes years to develop these programs. Other thoughts, kind of globally thoughts around, I've been doing public health and virology in this area for about 30 years. And I think many of my colleagues, and we've been screaming about this problem of not being aware of this coming in for the last 20, 25 years. I think, again, like HIV was a wake-up call, I think this hopefully is another wake-up call. And we have got to, as a country, figure out a strategy around national testing, stockpiling, algorithms of everything we've been talking about, reporting, stratified testing, so that when things like this happen, it's never going to be perfect, but we need a game plan. I felt like we were all playing our own game in every state, in some areas, in some cities, down to the point where it was just a mess at times. And I'm being critical of the U.S. and places I've worked at, like CDC and the Department of Health in Austin. We have got to learn some lessons, and hopefully we will do so. And then it's incumbent on us as scientists and physicians and healthcare professionals and academicians to continue the conversation in a way that the public raises health literacy through science communication. I am a total believer in this effort. I think we need more of us in places that are making policy, as well as trying to get people to understand these issues. Again, I want to thank the organizers of this seminar. I think these are very important tools and ways to leverage this information. And I also would like to thank my two colleagues for joining us. I've actually learned some things. I'll keep an eye on those tests that are coming out for the university. And I hope you guys will stay in touch as we move forward.
Thank you.
Thank you.
Dr. Crawford or Dr. Rohde?
Byron, you want to go ahead first?
Yes, I echo what Rodney said as well. The clinical laboratory scientists, medical technology is a huge shortage of med techs, hospital lab workers around the country. It's certainly a very important position now, because there are a lot of them retiring, and I don't see them coming up like we used to see. The school here puts out about 12 a year. There's a school in New Orleans that does about the same, and that's it for the whole state of Louisiana. I think master's or advanced degrees in public health would be helpful as well. I also want to bring up the physician assistants and nurse practitioners they're almost like physicians. I have a son who's a physician assistant in Pensacola. It took him 2 years out of college and to get a degree, and he pretty much worked as a physician there. The last 6 months since COVID, he's been running a COVID clinic, seeing patients that don't need to be hospitalized. So totally surprised that he hasn't developed COVID, but he takes all the precautions. There's 95 masks. I haven't seen him since last January, basically because I don't want to risk myself catching it from him. But that's another very important position: nurse practitioners and physician assistants. There are a lot of schools out there taking students now for PAs, nurse practitioners. So keep that in mind. You don't have to go to medical school to take it.
Great, thank you. Dr. Roy, closing thoughts.
Yeah, I guess the last words, I guess. So few things, I think a lot of you listening are engineers and tech person. So for you all, developing better tests, more accurate tests, more rapid tests, more scalable tests, more cheaper tests, that's an engineering problem. In fact, I'll tell you the NIH has invested over $100 million from NIBIB for testing all the tests. That's what we are doing at Georgia Tech right now. One of my colleagues, the PI of that, this is NIBIB. So every test that's getting developed, we test it about its sensitivity, its validity, its scalability, its cost, all of those things. The second thing is really working on the areas of therapeutics and vaccines. The therapeutics area has been very disappointing. I am almost scared and disappointed at that, because there is no good option for therapy once you get to a severe case. It's almost a Hail Mary at this point. So working in that space is very critical for years to come. This is not going to go away next year. It's not going to go away in 2 years. It's at some level will be there. And then vaccines work, and take the vaccine when it comes out and is approved, and advocate for people to take the vaccine. It is important as scientists, engineers, and other professionals to not just take it for yourself, but also advocate for taking it to others. Really, I think what this at least has taught me this last month, despite all the agony and hardship and scariness and all that, is if we all work together, we can accelerate development of drugs and therapeutics and diagnostics so much more faster than we have done historically. So hopefully going forward, those timelines that I showed in my first slide will change drastically. It won't be 10 months anymore after this, but it hopefully will not be 15 years. Maybe it's a few years now. And that pathway is now there. So with that, I hope lots of you start working in this space and the broader space. As a scientist, an engineer, as a technician, as a healthcare professional, any of that.
Science communication, yeah, all the areas. I think that's really important, that there's a lot of places for people to be involved and engaged in making things better in the future. So I just want to thank everybody for coming on and teaching us so much. It's been really great, and there's been a lot of very positive comments here. So thank you.
Thank you.
Thank you, Jamie. Thank you, Mary, and everybody. Bye.
Take care. Stay healthy.
Take care, everybody. Stay healthy.
Yes. Everyone have a great evening. Thank you.
You bet. Take care.
Chris, I have a question for Chris before you leave.
Yeah.